Welcome back. So we're just talking about the partial pressures of gases and how those are going to affect the movement of those gases. So now we'll see how that occurs in the inside in what we call internal respiration. So now that we have gases coming into the respiratory area and getting into the capillaries, now we need to get them to the tissues themselves. And so this internal respiration is going to occur at the tissue level. And so at the tissue level there, we're going to have the movement of essentially oxygen into the tissues and carbon dioxide away from the tissues. And so that process itself is going to occur as we move through the capillary bed that we have in the tissues themselves. And so on the arterial side of the membrane, we're going to have, or the capillaries, we're going to have a high PO2 coming in, and that's going to be higher inside the arterial blood compared to that of the systemic tissues themselves. And then the PO2 exiting there on the venous side is going to be much lower. And so essentially as we kind of move through the capillary bed here, we eventually kind of move through and change the PO2 that we have on either side there. And so on the arterial side, we're going to have a PO2 somewhere around 100 millimeters of mercury. And then on that venous side, we're going to get back down to somewhere in the neighborhood of 40 millimeters of mercury over there. And CO2 levels are going to be just the opposite. And so we're going to have a low CO2 level on the arterial side and then gain PCO2 levels on the venous side there. We've talked about before that in terms of our oxygen transport, hemoglobin is where it's at. And so this is where the majority of all of our oxygen is going to be bound to, is that hemoglobin. It's a very quick, rapid process. One hemoglobin molecule, again, can bind 4 oxygens. So there are 4 ions inside that hemoglobin molecule that can each bind a oxygen reversibly. And so when we have oxygen bound to that hemoglobin, we have what we call oxyhemoglobin. And so this process is going to allow us to bring that oxygen to the tissues. And then reversibly in the tissues, we're going to take that oxygen off of that hemoglobin, put it back into solution, and allow it to be delivered to the tissues themselves once it's there. The speed at which hemoglobin binds to or binds oxygen, or the speed at which oxygen binds to hemoglobin, is fairly quick. And so hemoglobin itself very quickly adds that oxygen to it. And when we get up to a PO2 of about 70 millimeters of mercury, we're going to find that the hemoglobin itself is essentially almost entirely saturated. So we have almost all of our hemoglobin bound with oxygen at that point. The process leads to essentially when we have low levels of hemoglobin, and we'll see this on a graph in just a little bit. And so as we move up towards that higher levels, this curve is going to basically be a steep curve, and then it levels off as we get closer to that 104 or so PO2 that the oxygen is coming into the lungs at. And so very, very quickly, and this whole process takes only about a quarter of a second for us to load all of that oxygen onto the hemoglobin itself. When we move through the tissues, for the most part, we typically think of, in a sense, kind of oxygen in and then oxygen out. And so, but in all reality, when we're coming to the tissues themselves, we're not going to unload all of the oxygen that's there necessarily, unless that tissue is really, really, really going through some metabolic activity, unless it's going through a huge amount of metabolic activity at that point, and then it's going to unload more of the actual oxygen that's there. So only about 20 to 25% of the actual oxygen that's bound is going to be unloaded at the tissues themselves. The majority of it isn't going to be unloaded at that tissue level unless it has a huge amount of necessity at that point. If we need more oxygen in the tissues themselves, we're going to have essentially a steeper curve once again. And so that curve is going to be in that level. So if the tissue level of oxygen is down around, say, 80 millimeters of mercury in terms of its overall average. That's going to have that oxygen come off of the hemoglobin much faster, and that's going to increase our ability to dump that off. At the same time, we're going to have to also increase respiratory and cardiac output in order to match the demands of that potential tissue at that time. So here we have our hemoglobin saturation curve itself, and this is going to be that area where we have kind of the greatest amount of binding in terms of the steep level of oxygen binding to the hemoglobin itself. Once we're there, when we're inside the lungs, we get up here to around 104 or so millimeters of mercury, and we have essentially complete saturation once it's there. In the tissues, we're going to unload down that 20% or so. So if we're at 100%, we're going to get it down 20 to 25% as we come into the tissues themselves. And we can see that we're getting into that steep part of the curve there where we can unload quickly into the tissues as well. But some things do influence how quickly those oxygen molecules are going to come off of the hemoglobin. Things like temperature, pH, and the amount of CO2 that's in the area. And truly, pH and CO2 are going to go kind of hand in hand with one another as well. And so as we change those levels, that's going to lead to a change in the tissue demand for oxygen as well. And so if you think about it, if we're using a particular organ, oftentimes the temperature is going to go up, which means that they're using more, the cells are doing more. So our PCO2 is going to go up, but then our pH is going to start to come down because it's going to become more acidic in the area as well. And so those are going to all have an effect on the amount of oxygen that's then going to want to come off. And so if we have that particular scenario, that's going to decrease hemoglobin's affinity for oxygen. So if we change that and make it so that our temperature goes up, our PCO2 goes up, our acidity goes up, and so we kind of have all of those go up, but with increased acidity, remember that the pH actually goes down. That's going to lead to oxygen unloading. It's going to go from the blood into the tissues, and we're going to have this change in oxygen leaving out towards the tissues themselves. And so that's going to allow us to dump more of that oxygen off into the tissues at that particular time. So if we have a change in temperature, and so our temperature becomes higher, there the affinity for the oxygen becomes relatively low. Same thing when we have a high PCO2, a low pH. We're talking about all those things that decrease the affinity for oxygen to hemoglobin itself on there. And then just do the opposite, come back into the other realm in a sense and lower the temperature, lower the PCO2, and raise the pH, and we end up having a greater affinity. Think of something like a tissue that is either not working or a tissue that may even be cold in times of things like even frostbite scenarios. So as cells do things, and this is what I said just a moment ago, as their metabolism goes up, and so as they metabolize more glucose, that's going to lead to more carbon dioxide being formed. That's going to increase the amount of hydrogen ion concentration that's there. So that's going to lead to a decrease in our pH as well. And that's going to decline there. And so a declining pH weakens even further our hemoglobin oxygen bond that's there. That declining desire for oxygen to be on the hemoglobin is what we call the Bohr effect. And as metabolism goes on, as we break bonds, that gives off heat. So our temperature is going to start to rise in those particular tissues themselves as well. All of that leads to more and more oxygen unloading. And so we're going to dump it into the tissues because the tissues now have an increase in their metabolic rate. Carbon dioxide moves in a somewhat similar process in terms of its movement. The big difference that we'll talk about in just a little bit is that more of the more of the carbon dioxide is not bound to hemoglobin. So it doesn't have a ton of it bound to there. And so we'll see here that we have as much as 10% just simply dissolved inside the liquid of the plasma. Only about 20% is carried in the red blood cells as bound to the hemoglobin. And when we see that, we have what we call carbaminohemoglobin. Nice little fun word to say there. Say that 5 times in a row very quickly, and that becomes a little bit of a tongue twister. And then the majority of it actually goes through the red blood cells and becomes bicarbonate. And so we have bicarbonate here, and it's going to go through the red blood cell, end up back into the plasma itself, and we have this bicarbonate transporting through the blood. And that becomes part of our bicarbonate buffer system that we have in the body itself to be able to buffer the pH that we have within the body as well. So carbon dioxide basically goes into the red blood cells. Inside the red blood cells, we have water. And so as water combines with the carbon dioxide, we get a substance called carbonic acid. That carbonic acid is now inside of the red blood cell, and it's going to then lose a hydrogen ion and the bicarbonate. So now we've lost that hydrogen ion. The bicarbonate can now move into the plasma. And so we're going to have this process going back and forth and back and forth. In order to do so, we do need an enzyme present, and so that enzyme is carbonic anhydrase, and that's going to catalyze that movement of carbon dioxide and water to the carbonic acid itself. So this process that we just described is this one here, going in this direction, and that's going to lead us to move in the direction of taking carbon dioxide out of the tissues, out of the plasma, and take it through the red blood cell. We have carbonic anhydrase inside that red blood cell that converts us into our carbonic acid. We dissociate one of the hydrogen ions off, and that bicarbonate ends up out here in the plasma. Since we just moved a negative ion out into the plasma, we're going to get a negative ion moving back into the red blood cell, and this is what we're going to call the chloride shift. So we're going to get a shift of chloride ions into the red blood cells as those bicarbonate ions move out. And then the process itself is going to basically be reversed as we move into the lungs as well. We also have here the movement of, so our oxyhemoglobin. The oxygen dissociates from that of the hemoglobin, and that then allows oxygen to move into the tissues themselves. And that's going to be the major way that the process occurs. So bicarbonate quickly diffuses from the red blood cells into the plasma. Our chloride shift, as I just mentioned, we've got to balance out that negative ion from the bicarbonate moving into the plasma. So we're going to get chloride ions moving back into the red blood cells or our erythrocytes, and that's going to allow us to keep a charge balance between the inside and the outside of those red blood cells. At the lungs, basically everything just goes in reverse. And so bicarbonate ions move back into the red blood cells. They bind with the hydrogen that they, in a sense, left behind before to form carbonic acid. We then have that carbonic anhydrase once again, and that's going to then split our bicarbonate or our carbonic acid back into carbon dioxide and water, and then that carbon dioxide is going to move along its concentration gradient and move into the alveoli of the lungs themselves. And so we just reverse this process to move our CO2 back in. A little bit of carbaminohemoglobin. We didn't talk about that on the last picture slide there. So a little bit of carbaminohemoglobin moving through the red blood cell that way. We have another 10% or so of just CO2 floating inside the plasma itself. And then, of course, there is CO2 and water in the plasma as well. But here we don't have the benefit of that carbonic anhydrase. So this is a slow process in order to move that small amount back into the lungs as well. And so now we've dumped off all of our carbon dioxide. We've picked up oxygen. So once again, oxygen moving in, following that pressure gradient, binds to the hemoglobin almost entirely. The degree of carbon dioxide transport that occurs, and essentially the amount of oxygen that stays on that hemoglobin, is dependent a little bit upon that of carbon dioxide. And so when carbon dioxide affects the hemoglobin saturation, this is what we call the Haldane effect. And so the lower the amount of oxygen that we have, and the hemoglobin saturation with oxygen, the more carbon dioxide can be carried in the blood itself. And so essentially, as our oxygen levels fall at the tissues, so as our O2 levels go down, that means that CO2 levels coming back into the blood can start to rise. And so that means more of that carbon dioxide can be carried in the blood itself. And this is essentially that Haldane effect. And so as more oxygen gets delivered into those tissues, the more CO2 can then get picked up by the blood itself. And so as at the tissues there, more carbon dioxide enters the blood. We have more oxygen being dissociated from the hemoglobin. More of that carbon dioxide will start to combine with the hemoglobin itself. But again, we're only going to transport about 10% that way. That's there. So, but the more that the carbon dioxide gets into the blood, the more bicarbonate we're forming inside there. And that's also going to help to take that lower pH away from the tissues themselves as well. And then we just simply reverse the whole process once we get into the lungs as well. Mentioned that the bicarbonate can have the ability to buffer the blood as well. And so we have this bicarbonate buffer system. So carbonic acid bicarbonate buffer system is going to make it so that as we look at essentially pH levels, those pH levels have, in a sense, kind of just very little changes. We don't want to have extreme changes in pH. So those pH levels, we want to stay relatively flat as far as the overall levels go. And so we're going to attempt to maintain that with the ability to have carbon dioxide buffered inside the blood itself with that bicarbonate itself. So as hydrogen ion levels go up, so if we have an increase in hydrogen ions, that means that our pH is falling. So increased hydrogen ions, decreased pH. The tissue or the blood is becoming more acidic in that sense. And so we're going to combine that together and allow us to buffer the blood at that point. And that's going to allow us to maintain that pH at a relatively constant level at that time. So excess amounts of that hydrogen ion are going to be essentially combined with our carbonic acid. We're going to get our bicarbonate, and that's going to allow us to buffer the system itself. As those hydrogen ion levels then drop, carbonic acid then dissociates, and we release more of that hydrogen ion to bring it back up. And so it's just simply that either binding up the hydrogen ions themselves, and so they're going to get removed, or we're going to release them and bring the pH back down as well. So carbon dioxide has that effect on the blood. So just simply doing something like holding your breath. So as we stop breathing in, CO2 levels are going to start to rise. That means that our pH is going to start to fall. That means that we're going to have an increased need for our bicarbonate in order to buffer this system out, and so in order to alter that. If you do hold your breath for a period of time, essentially your first reaction once you stop holding your breath is going to be an increase in your breathing rate. And so we're going to have an increased breathing rate in order to try and get rid of that CO2, which is then going to reverse our pH and start to bring that back up. And with that, we then have now, again, had a respiratory compensation for that increased CO2 levels that we had in the blood. Next up, we'll continue to look at the respiratory system and move on into the factors affecting respiration itself.